I wish to welcome all of you to this, the 34th Annual Arthur Sweeny Jr. Lecture. Our speaker this year is Dr. Martin Chalfie, who is the William R. Kennan Jr. Professor of Biological Sciences at Columbia University. In 2008, he received the Nobel Prize in Chemistry together with Osamu Shimomura and Roger White-Zien for the discovery and development of the green fluorescent protein, GFP. In addition to winning the Nobel Prize, he has received over the course of his career numerous honors and awards, including the E.B. Wilson Medal from the American Society for Cell Biology, Distinguished Scientist Award from the American Heart Association, and 2 honorary doctoral degrees. His talk is entitled GFP, Lighting Up Life, and will cover the story of the discovery of green fluorescent protein and also show the importance of basic research on non-traditional organisms. It is my pleasure to introduce to you the 34th Annual Arthur Sweeny Lecturer, Dr. Martin Chalfie.
I hope you can hear me. Thank you very much, Mark, for that very nice introduction. I want to say just one thing. I read online the information about Professor Sweeny, and I really like that I've been asked to give this lecture that actually honors someone who is really dedicated to teaching, which I think is exceptionally important. So I'm very grateful for the invitation. And Andre, thank you for all the things that you've done in helping me get here. So what I want to talk about today are the experiments that were involved in my involvement with this rather remarkable protein, GFP, green fluorescent protein. But I want to start in a slightly strange way, because I want to talk about, because this is also going to be a story, I think, about how people do science. And what I'm going to tell you is that when I was growing up, I learned a lot of things about scientists because of all the stories that were told about the great scientists of the past. Galileo and Newton, Einstein, Fleming, Darwin, all these great scientists of the past. And they gave me an idea about what it meant to be a scientist. I didn't have a lot of time to tell these stories, so they were told rather quickly, but they gave, and none of the things I'm going to tell you now were things that I actually was taught specifically. Learn this, it'll be on the test. But they were all things that I gleaned from hearing about these great scientists of the past. And I want to show you what those things are. And then I'm going to show you, I'm going to tell the GFP story, which basically says all of those things were completely wrong. Okay? So what were these things that I learned in the 1960s, 50s and 60s when I was going to elementary and high school? The first thing is that scientists are geniuses. Right? There's something innate about them. They are just a special breed apart. They are unbelievable. We've never seen anything like them before, and that's why they're scientists. And if you are not one of this club, then forget it. There's nothing you can do about it because you've lost your chance. And it doesn't seem to work on it. It's just something that was innate. The second thing is, because they're these geniuses, their experiments work all the time. Right? We never hear about anything going wrong. It's just Newton had this idea about the apple and then did some experiments and they all worked and this was wonderful. And this is how science is done. We tell these short stories about the accomplishments. The next thing about scientists that I got from all the examples I was given is that scientists, because they're geniuses, use a very peculiar way of thinking called the scientific method. They come up with a problem, they think about that problem and come up with a hypothesis. Once they've come up with a hypothesis, they figure out the perfect experiment to test that hypothesis. They do it, the experiment works the first time, and all the money rolls in. So they're purposeful in what they do. They actually know what they're doing at all times, and that's what it means to be a scientist. The other thing is that until, and this was not really until sort of the middle 1960s, sort of late, early, like 62, 63, in high school, we didn't know about Watson and Crick. And all these examples of scientists that we ever heard of, they all seemed to work alone. They were individual. Now, they usually had an assistant named Igor. But other than that, they really didn't have anybody else that they worked with. They were always working on their own. And the final thing about all these people, except for one person, and that was Marie Curie, every single one of them was a white man. They were European or of European descent. Every single one of these scientists. That was it. That's what it meant to be a scientist. And I'm going to tell you a story that actually, as you haven't already figured this out, all of this is complete nonsense. My involvement with this wonderful protein that comes from the jellyfish Echoria victoria started in 1989. I had had my lab at Columbia for 7 years. I had done a postdoc before that in Cambridge, England, and I had been working on this wonderful animal, actually the only animal anyone should ever work on, this small roundworm nematode Cenorhabditis elegans. And this was important for this story because from 1977 until 1989, when the story really picks up, I had been giving talks and telling people about how this animal was so wonderful, all the genetics we could do, all the stuff we knew about it. And every time I gave the talk, I would say one of the wonderful aspects of this animal, it is transparent, okay? So by 1989, even I believed that that was true. I had said it so many times. What I really work on, what I worked on before GFP, after GFP, what we still work on now, is the sense of touch. I'm interested in the general problem of how we interact with the world around us. How can we detect the world around us? Scientists have known, biologists have known for the last 130 years how we can interact with the world around us if the interaction is through light. Because we've known for 130 years about the molecule rhodopsin, which is in our retina, that allows us to detect light. And we know a lot about its molecular basis of how that sensory detection or transduction occurs, how that leads to changes in the electrical properties of the cells, and then eventually signals are sent to the brain. And we've known for about the last 25 years or so how we interact with the world if the signal from the outside world is chemical. So we know about receptors that are needed for taste or for olfaction, and internally we know about chemical receptors for hormones and neurotransmitters. And we know a lot about how these work at the molecular level. But we have a vast number of senses that don't work because they're activated by light or by chemicals, but they're activated physically. They're mechanically gated, opened, or activated, and these senses are what underlies our sense of touch. We have something like 5 different types of cells in our skin that respond to different types of touch. We have hair cells in our inner ear that respond either to sound for us to hear or acceleration, the vestibular apparatus. We have cells that sense the stretch of our muscles and our tendons. We have cells that detect blood pressure, and there are several others as well. Now, all of these mechanically driven senses have one thing in common. We have no idea how they work. This is a good area to get into because it's wide open. We really don't understand the molecular basis of hearing or touch or balance or stretch of our muscles. And so that's what I work on. And I use this animal to do this. This is a very sophisticated experiment. We take animal is only a millimeter long, 25th of an inch. Everything's done under a microscope. We take a toothpick and we glue an eyebrow hair to it, because eyebrow hairs are usually nice and tapered, and we tickle the animal. If we tickle the animal in the tail, that's the tail, it moves forward. If we tickle it in the head, it goes backwards. And if we give chemicals to cause mutations, we can find mutants that don't respond to this tickling. Now, you may have realized that dead animals also don't respond to this. So we actually have a different way of thinking about this, which is we show that they have normal movement, but they just don't respond to this gentle touch of a hair. And we've collected hundreds of mutants like this, which I don't know whether that's an inducement to come to my lab or not. But we have, in the course of the experiments, by 1989, we were starting to clone the genes that were mutated in our various animals, because we wanted to understand, can we actually find the sensor for touch? And I won't go into that, but we actually did. That's a completely different set of experiments. But we were starting to clone genes, and when you start to clone genes, one of the first questions you want to address is, what cell activates that gene? Where is it turned on? And in 1988, 1989, there were a number of ways to do this, because you know DNA makes RNA, makes protein. So one way is to simply decide, and it's a little hard to see here for the light, but if you look here, you see there's red lines here. That's making an antibody against the protein and seeing what cells make the protein. That'll tell you where the gene was active. Or we can use what's called a reporter system. In this case, the enzyme beta-galactosidase. We can use a substrate, the substrate called XGAL, and when the enzyme sees the substrate, it makes a blue deposit, which you can see here in these various cells here. Only the gene that turned on this thing will allow that blue color to be made, so we'll know where the gene was active. Or we could look directly at whether RNA was made from the gene by a process called in situ hybridization, where we take a DNA probe and use that to detect where it is. And you might just be able to make out the dots here in these cells. So all three of these methods answer the question, what cell turns on the gene? But there were a lot of problems with these experiments. One of the problems was it took a lot of work. We'd have to take the animals, fix them, which really meant killing them, and then stabilizing everything so nothing got all jumbled up. And then because we had to get the antibody into the cells, into the specimen, or get the enzyme substrate into the reporter system, or get the DNA probe for the in situ hybridization, we had to permeabilize the sample to get things in. Well, so we've killed it, we've fixed it, we've permeabilized it. This is going to give us a snapshot in time. We're only going to be able to get a very quick glance at that instant when we killed the animal. If we wanted to look over time at how things changed, we'd have, first of all, if we just wanted to see it happen again, we'd have to do the whole procedure all over again. But then if we wanted to see how it changed over time, we'd have to do time points and do all the preparation for animals at different ages, for example, or after various times after a treatment. And all of this was a lot of work. And again, it's because we had this very static view of what was happening. Now, before leaving this slide, I want to explain my color code, because I have a color code for all of the pictures of people that you're going to see and their names. So these two people, Hongping Du was a graduate student in my lab, and Shohei Matani was a postdoc in my lab. Anyone who worked in my lab, their name is going to appear in red, so you'll know that. Anyone whose name appears in blue is a collaborator. So it's not from my lab, but someone we collaborated with on the project. Anybody whose name is in black did something I wish I had done, okay? And one of those people was what started me on this whole thing, Osamu Shimomura, who shared the prize with me in 2008. I went to a seminar in 1989, and the speaker started talking about Osamu Shimomura and his work on this jellyfish, Aquaria victoria. If you have 29 minutes to spare, and I would urge you to spare them, please go to the nobelprize.org website and watch his acceptance speech. You can ignore all the rest of us, but watch his speech. His story is amazing. At the age of 16, he's told, sorry, you have to quit high school and work in a paint factory. So he quit school, He goes to work in the paint factory. Now, the paint factory is outside of the city he was born in, so he had to go over the mountains to the adjacent valley and work there. But this turned out actually to be a good thing because the year was 1945, and the city was Nagasaki, Japan. And he was on the other side of the mountains when the atomic bomb went off. He saw the flash. He went in the next day and tried to help people and rescue them. But what it meant is there was absolutely no place to go to college at all, because people usually went to the local college and everything had been destroyed. After several years, he finally gets a chance to go to college, but the only college that he can go to is one he wasn't particularly interested in. Nothing against it, but he just didn't want to go to pharmacy school. But he goes anyway, and he gets his degree, and goes to work in a job at Nagoya University, working as a technician. Now, as he goes and works as a technician, he's given something that, this is sort of one of those secrets maybe I shouldn't tell, but he's given one of those projects we often give to students. It's a project in which we know we've given this to several other really bright people, and it has not worked. But the new person doesn't know that. And so let's give it to him or her. And so we give this project to this person and say, okay, see if you can get this to work. Except in his case, he got it to work, because it was about a small crustacean called Cyprinina. And the problem was, it was known that Cyprinina is one of the many organisms that can actually generate light. You know, of fireflies, there's Cyprinina, there are glowworms, there are jellyfish, which I'll talk about in a moment, there are bacteria, there are fungi, there are many, many different types of organisms that can generate light, which in itself is a fascinating biological problem. How do they do that? Turns out they all do it differently. So knowing what one does doesn't really help you on the next. But he was able to figure out this one from this small crustacean. That led to two things happening to him. The first was he was invited to go and do some more research on bioluminescence, as this is called, in the United States. And the second thing was his boss at Nagoya University arranged a rather unusual going away present for him, his PhD. So this is Osamu. He comes to the United States, and he goes then to Friday Harbor Laboratory in the state of Washington and starts to work on trying to figure out how this jellyfish, Aquaria victoria, produces its green light. And he goes and he's working on this. This is the part about the scientific method, in case you're taking notes. He does the experiment, it fails. He does it again, it fails. He changes the conditions, it fails. It fails, it fails, it fails. The whole summer, things do not, it's using up a lot of jellyfish. Nothing is coming out of this. One day, he's done the prep. It's late into the evening. He's tired. I think he wants to go home for dinner. And it's failed another time. So he's a sort of neat guy, and he goes and cleans up after himself, and he throws the prep, which is now worthless, in the sink. Now the sink had, probably some jellyfish parts in it, and had a lot of seawater because they had tanks that had seawater in it that were emptying into the sink. He just throws the stuff in there, turns off the light, and is just about to go home when he happens to look at the sink, which is glowing brightly. This is a surprise. Nothing glowed brightly before. He goes to the sink. He starts to think about what it was that was there. And he says, seawater. Seawater has calcium. I never added calcium to the reaction. And the next days, he adds calcium, and he finds that's the thing that's missing. And so this is the scientific method. If you can't get it to work, throw it in the sink, throw it on the floor, do something like that. But the upshot of that is he isolates a protein in 1962, which he names after the jellyfish and calls it aquarin. And that aquarin plus calcium produces light. So it's an accident. completely accidental discovery by throwing it in the sink. And then he looks at it, and I guess he must have been a little depressed, because he looks at it and he goes, the jellyfish produce a green light. This is blue. Wrong color. That must have been very frustrating. But he thought about that for a little while and said, wait a minute, maybe there's something that converts the blue light to green light. Now, if you have a molecule that accepts light of one wavelength and gives off it at another, that's fluorescence. And so he went with a handheld ultraviolet lamp, and he went to all the samples, and he said, oh, look, here's a different sample entirely, and if I shine this light on it, I get something green coming back. And so he writes this paper in 1962, and he says, in footnote number 3, he says, in addition, there's this other protein that actually has the right color that can be activated by a handheld UV lamp. I'm going to call it the green protein. Now, we, of course, now call it the green fluorescent protein, or GFP. I believe this is the only footnote in history to get somebody a Nobel Prize. But it did. And so with aquarin, calcium, and now this protein, what's generated is a green light. Now, to set the stage, it's 1989. I've been working on a transparent animal, and I want to know where genes are expressed, and I have to go through all this rigmarole, and I suddenly realize if I could get the genes to make GFP, then all we'd have to do is shine blue light onto the molecule, and we'd see green. In effect, that it was serving as a lantern, and we'd be able to see things. And I got really excited. I got so excited that I cannot tell you what the seminar was about. I can tell you the introduction, which I just did, but I don't remember any of the rest of the seminar, because all I could think about were the experiments I wanted to do if this thing really existed. And I thought this would be just great. What I want to tell you I didn't do, maybe I should be embarrassed about this, but at least it's the truth. I did not sit down in my lab and say, we should have a better marker for looking at gene activity. I wonder what would be a better marker. I got an idea. That marker should be a fluorescent protein. I wonder if there are fluorescent proteins. I'll go look for. I didn't do any of that. I sat there passively. I work on a transparent animal. I was looking at gene expression, and this guy gives me this gift of a seminar by just talking about this work. Now, there were lots of other people in that audience. And as far as I can tell, absolutely none of them then or at any of the other times had this idea. I think the reason I had the idea is because I happened to also be working on a transparent animal, and not frog or mouse or some fruit fly or something like that. And I was interested in when genes were active. So it was just the right time and the right place to do this. And so I was very excited. And I found out that this man, Douglas Prasher, was in the process of isolating the DNA for green fluorescent protein. And I got in touch with him, and we decided we were going to collaborate on the experiments, which we did, but unfortunately for 3 years we lost track of each other. Then in 1992, Gia Skirkin came into the department as a beginning graduate student. She had just finished a master's degree in chemical engineering in our engineering school at Columbia, and she had worked on fluorescence. So to me, she was perfect. Right? She worked on fluorescence. She would be interested in this, and I told her about the idea, and I said, but Douglas Prasher has never gotten in touch with me. I don't know what's wrong. It probably didn't work. Turned out, I thought he hadn't succeeded, and he thought I had quit science, because we could not get in touch with each other. But we were able to look online, and there was his paper. And we went down to the library, pulled the paper out in the library and looked at it, and the paper had something that most scientific papers do not have. It had his new phone number. Called him up, collaboration was back on. 3 to 4 days later, he sent the samples to us of the DNA, and Gia started to do experiments to put it into bacteria. Now, this is a little bit in support of those nonsensical things that I said at the beginning, because her very first experiment worked. And that turned out to be exceptionally important, as I'll explain. And this page from October 13th in 1992 is that experiment where, for the very first time, GFP worked in the animal, or in bacteria. It's circled there. It says strongly fluorescing E. coli. She put it in bacteria, and she took a picture of those first bacteria with GFP in them in this, that I show also on the slide. But also on this slide is another area I'd like to point out. Again, how science may actually be done. It says there that she used the scope from engineering. This is the microscope in her previous laboratory where she had gotten her master's. And the reason she used that microscope is she tried to look at the samples on my microscope and rapidly realized that my microscope was a piece of junk, and that she wasn't sure if it worked or not, but she knew where a better microscope was, and so she went to that one and then was able to take the pictures. Now, we used that microscope a little bit after this, and we used other microscopes in the department, but eventually we got kicked off of all of them. And so we had a problem. We needed a microscope. The way I solved that problem is I called up all the sales representatives of microscope companies in New York City, and I said we had just discovered a new way of looking at gene expression, and I was very excited about the new method, and I was going to buy a new microscope. But before deciding which microscope I should buy, I needed to try them out. So if they could bring it by and let us have a trial on them, and probably a good trial would be if they could leave it for 1 or 2 months, then we would be able to know where to buy it. And so all the experiments we did were on borrowed microscopes. Eventually we did buy one, but not until after the paper was published, actually. Now, why was it really important that Gia's experiment worked the first time? It has to do with what the people working on GFP at that time knew about the molecule. In its favor, as a way of marking cells, was the fact that it didn't need to have any extra cofactor added with it. All of those was needed was the protein itself. So that was good. Nothing else was needed. What was bad is that this protein undergoes a rather unusual change. So if you look here, this is part of the protein, and I've drawn it in a slightly strange way. Here is the peptide backbone, so serine 65, tyrosine 66, glycine 67, and then on down the polypeptide. And the mature protein takes these 5 atoms here, and there's a condensation reaction, and you wind up with this 5-membered ring, which is absolutely needed for the fluorescence. No one had any idea how this was made. They thought there had to be a converting enzyme, something that caused this reaction. Maybe 2, maybe 5. Well, if you need extra things, this is not very useful, because then you're going to have to add all those things to get it to work. So we decided, oh, what the hell, let's just put it in and without anything extra and see if it works on its own. And the fact that it worked the first time on its own said there are no converting enzymes. It is its own converting enzyme. It allows this condensation to happen when the protein folds correctly. So you don't need anything else. So this was an exceptionally important experiment, not only to show that it works, but it works without needing anything else. So it'd be a very useful tool. It turns out that there were 3 other groups that had the same idea to do this experiment. All 3 of them did the experiment, and for all 3 of them, it did not work at all. Worked for us, didn't work for them. What was the difference? The difference was sloppiness. So this is going to be a little plea for sloppiness. Douglas's clone, which I've diagrammed here, and it's actually probably better to just look down at the bottom. His clone that he got from the jellyfish encompassed everything including the red bits and the green bit. The green bit is what actually codes for the protein. The red bits are extra jellyfish stuff. Okay? At the time, 1992, there were two main ways, well, we still use these, two main ways to generate a lot of DNA. One way is to let an organism grow it up and then take a restriction enzyme to cut that DNA out. And it's going to be perfect, or as perfect as the organism can make it, because they're just copying it over and over again. So you're going to get lots of good copies. But you're going to get the extra bits. I didn't like having extra bits. I just inherently didn't like to have anything extra. So I suggested that she use polymerase chain reaction, PCR, to amplify it. In PCR, you can, of course, take primers, these little green arrows here, that just will copy the coding sequence and not the red bits. And that's what Gia did. Why? But at that time, PCR was known to be very error-prone. Lots of the stuff that was made had mistakes in it. But remember the type of experiment that we were trying to do. We were going to take the DNA and we were going to put it into millions and millions of bacteria. We didn't care if half of the bacteria had bad stuff in it. We just wanted one bacterium to have good stuff. So I was much more enthusiastic about accepting this not working in a lot of cells to have it work in some of the cells, which I showed you the picture it did. So mistakes were okay. Sloppiness was okay. Having extra bits that I knew nothing about, I was scared of those. For good reason. It turns out, and no one really knows what it is, there's something in those red bits that makes it not work. But remember, you would conclude, it doesn't work. It must need a converting enzyme. And then you'd become very unenthusiastic about the experiments. But for us, having Gia's thing work the first time said there was no converting enzyme, and it worked. And so we started doing things. Among them, we put them into worms, and it worked great in worms. And so we decided we would publish. So I want to say a little bit about scientific publishing, some of the problems, some of the pains that you get. We were able to publish it. We published it in the journal Science because we really wanted a very broad audience to look at this, not just the cell biologists or the developmental biologists or the neurobiologists or the geneticists, everybody. We wanted everybody to look at this, so we didn't send it to a specialized journal. Now, some of you may know that the journal Science, the people that work on the journal Science, think very, very highly of themselves and the journal. And as a result, they don't just send your paper out for review. They think about whether your paper is worthy enough to be sent for review. And they called me up and they said, we're sorry, we're not sending your paper out for review. And I said, why? And they said, well, to tell you the truth, we don't like your title. And I said, you gotta be kidding. And they said, no. And I said, what's wrong with this title? Green fluorescent protein, a new marker for gene expression. And they said, well, you see, everything that we publish in the journal Science is new. You cannot use that word in the title. And so I said, if I change the title, will you send it out for review? And I think they actually said maybe. I'm not really sure. And so I wrote a new title. I have to admit to you that I really do not like to be told what to do. I think that's what I enjoy being a scientist about. And so the title I sent in was probably not the title they were expecting. It was the following. The Acora Victoria green fluorescent protein needs no exogenously added component to produce a fluorescent product in prokaryotic and eukaryotic cells. Now, I could have just had the figures in the title and that would have been the end of the paper. It's all there in the title. But they reviewed it, the reviewers liked it, they suggested it be published, and then the copy editor called me up. And she said, well, we're setting up your article for publication, and you know that the title is a little long? And I said, oh, would you like me to shorten it? And she said, oh yes, we really would. And I said, well, I'll try to think about how I could do that. How's this? Green fluorescent protein as a marker for gene expression. Oh, that's wonderful. And that became the official title. The second problem we had with publication has to do with this picture that was on the cover. This is a picture of a newly hatched worm, and it shows, among other things, this nerve cell growing out. So that's the growing end of a nerve cell. So I wanted to point out that you could watch living organisms and watch things happen because of this marker. So I thought this was a pretty good way of advertising what it was. The cover editor calls me up, and she says, you know, we really like your picture, and we really want to use it on the cover. I said, that's great. She said, we have a problem. I said, what's the problem? She said, there's one color that doesn't work on the cover. We tried to do anything else. Can we change it? It's green. We can't do green very well. And I said no, and this is actually a little enhanced here. So if you actually go and look at the original cover, it really looks washed out and horrible, but I couldn't imagine them doing another color. So there's two. There's one more problem. I'm a blabbermouth, right? I like telling people about what we've done, and so we had already told lots of people about GFP way before we published it. And we were sending out samples to people. And some of those people that worked on yeast or dictyostelium or Drosophila, other organisms, human cells in culture, had already gotten back to me and said, it works in our system. And I wanted to put that in the paper. And I asked them, can I have permission to cite your unpublished work in our paper? And they said, you gave us the stuff before you published. Of course you can. That's perfectly all right. Except for one person who made a series of excessive demands. This person. You probably can't read them, so I've blown up the important part of this. I had to make coffee, or I was supposed to make coffee, for 2 months every Saturday morning. I was supposed to prepare a special French dinner and take out the garbage nightly for a month. This is my wife. But Tula did something that was exceptionally important. Because, you know, genes, to be very simplistic about them, genes have two parts. They have the part that encodes the product, the RNA or the protein that's being made. And then they have all the instructive parts, the control, regulatory parts that say where, when, and how much of that product should be made. And so what we did in our experiments was we took that control part and we put it with GFP. So now it was driving GFP in the right place and time and so on. What she did was take the gene she was interested in, Drosophila and fruit flies, and take the control region, the part that was needed to encode the protein, and then GFP. So now when the protein was made, it had that lantern hooked up to it, and so it could move all through the cells. And this is shown here. This is the developing oocyte in Drosophila, and the protein she was interested in is now labeled with GFP. It comes through these canals and comes to lie on this surface of the oocyte and at this surface. And she could watch that happen. She could watch the protein move in and assume the right positions in the animal, in the cell. And so this was very exciting. It was the first protein fusion, the first protein hybrid with GFP. So it was an exceptionally important experiment. It was really wonderful. She says, I've never paid up. It's a debate in our home. So what's important about GFP? The thing that made GFP so exciting as a tool to use is that the protein was encoded by DNA. So if you could get the DNA in whatever organism or cell you were interested in, it would become incorporated into that DNA, and that cell or that organism would generate GFP, and its progeny would, and its progeny, and so on. You could establish a line that could do this. So its DNA could be inherited. You didn't have to do the experiment once and then do it over and over and over again. You did it once and then just looked at the subsequent organisms. Looking at GFP, we didn't have to kill the animal. We didn't have to fix the animal. We didn't have to permeabilize it. All we had to do was shine blue light on it. And wherever the blue light hit the protein, we would see green. And that was it. Pretty innocuous. Fairly non-invasive. makes you think that what you're looking at is actually real biology. It's a relatively small molecule, and that means that unlike beta-galactosidase, which really stayed in the cell body, GFP could go to anywhere in the cell. And for nerve cells, it could just go absolutely anywhere in the cells. And finally, as I've said several times, you can actually look at living tissue, living organisms, and see what was happening over time, because we could actually see life happening in cellular detail. So some examples of organisms with GFP. So we have our worms. This is all the nerve cells in the animal with GFP expressed in them. This is a canola plant, fruit fly, mice, zebrafish. Actually, you can buy these in pet stores. They're called glowfish. And this is Alba. Alba is the GFP bunny. The artist Eduardo Koch commissioned a French biotech company to generate Alba, who was the family pet. Eduardo would go to different art shows that he would have and bring Alba to stimulate people talking about the connection between art and technology and art and science. And Alba was the family pet and lived to a ripe old age as far as rabbits go, but is now no longer with us. On the right here are several cells from different systems. We have a mouse fibroblast. These are some cells from Drosophila, fruit flies. This is from the mustard plant, Arabidopsis thaliana. And this is a mouse Purkinje cell, and you can see how the GFP has really outlined all the different processes in this cell. Now, I keep telling you you can see living things, so I want to show you two very quick movies. One of them has GFP attached to a major protein, beta-tubulin, for the spindle. So when cells divide, or in this case it's going to be early embryos of Drosophila, so it's going to be nuclei dividing, there are no cells. The nucleus breaks down, a spindle forms, the chromosomes attached to it are separated, and then you get two new nuclei as they reform. So the spindle will be seen during cell division. The other movie I'm going to show you has a small peptide attached to GFP, which is called a nuclear localization signal. What this does is anything that's attached to it gets sucked up into the nucleus, if there is a nucleus. So before division, all the GFP is going to be in the nucleus. During division, it's going to go all over the embryo, and then when the nuclei reform, it's going to go back into the nucleus. So let's see these movies. The one on the right, both of them were by a graduate student in Canada who I've never met, but she donated these to the American Society for Cell Biology website. Her name is Rosalind Silverman Gavrilla. Here is an embryo, a Drosophila embryo, and these are all very speeded up. Okay, cell division does not go this fast, but at least it allows you to see several things dividing. So spindles form, come apart. Now we're going to get the next round of cell division, and then the next. And I think there's going to be one more, and then it's going to cycle through. And the one thing that should strike you about this movie is that everything is happening at the same time. In fact, she calls this movie In Synchrony. And so it immediately asks the question, how does that happen? What are the controls? We know a lot about what controls cell cycle. I'm not going to go into that now, but it's being able to watch these things in living organisms that starts to introduce questions that you never realized previously. Now, on the right-hand side here you see colored nuclei. This is false color. The convention is if you have a little bit of GFP or a little bit of anything, that's at the blue end of the spectrum. A little more, you get green, a little bit more yellow, then orange, then red. So this is blue, this is green, these are sort of yellow-orange. This is sort of green up here as well. So different amounts of GFP. She names this after the Van Gogh picture Starry Night, and you'll see why as I show this movie. So now it's all over the cell division. The nuclei are going to reform, And as they reform, the GFP is taken into them because of the nuclear localization signal. Then the nuclei break down again, and the GFP goes all over the embryo, and it's going to go through this several times. But as you look at this, you see it's not synchronous. It's actually going as a wave from one part of the embryo to another. But this is the same sort of embryo. Why is this happening? We actually think this is a sign that the embryo was unhealthy, probably smashed a little by the cover slip. So there's a little more pressure at one end than the other, and that was causing the problem here. But it's a very sensitive way of looking at the health of the system by how the cells are dividing. But it also makes a great movie. So GFP has been, I talked to people in chem abstracts, and they said that they actually record every single word that's been published. And I said, does that mean you can tell me how many papers use GFP or other fluorescent proteins since our paper in 1994? And they said yes, and they got back to me, and it was around 160,000 scientific papers, which is sort of mind-blowing. So GFP has been used in a lot of different ways. I'll talk about them as fundamental research, applied research, and unexpected research. What do I mean by fundamental? Well, it allows us to look at gene expression, protein localization, as my wife did. We can mark cells and then ask, can we find mutants in which those cells grow in the wrong place, or there's the wrong number, or there's something different about them, and then use that to study those processes. We can label cells and tissue. We can actually use that to isolate cells. We could look, as people have, at brain circuitry by labeling the nerve cells. We can look at interactions, host versus parasite interactions or so, asking a lot of basic fundamental questions about biology. But in terms of applied, there's lots of things having to do with human health. You can make cancer cells have GFP in them, look at them in mice, we're not talking people here, and ask about how the cancer cells metastasize from one place in a body to another. We can ask how HIV gets distributed, how the virus gets from one cell to another. We can talk about inherited diseases, infections. It's been used in biotechnology, interesting. It's been used for drug discovery, and it's been used for biosensors. And I'll give you some examples, one of HIV and one of the biosensor in a moment. And then there's what I would call the unexpected, things that I had no idea about. Some of these things I sort of thought were going to happen, but others were just strange. There's been a lot of new research. Once GFP came out, people started saying, green's nice, what about other colors? So Mikhail Nats, Lukyanov, I forgot his first name, in Russia, in Moscow, these guys loved to go scuba diving. And they knew that corals were fluorescent. But some corals were fluorescent and gave off red fluorescence. And so they discovered the first red fluorescent protein by going and getting the gene from the corals. But they wouldn't have done it without GFP. Completely unexpected. And now there's many different variants of fluorescent proteins that come from all sorts of organisms that people have gone off to get. The whole field of optogenetics, we're shining light on cells that have particular added genes to them, actually can either get the nerve cells to work or to stop working. Some of the people that were involved that actually came to me and they said, you know, we wouldn't have done this work at all had GFP not been there. So we realized there were a lot of other useful tools in other organisms, and that's why we went off to do these experiments. And there's been, people have looked at new chemistry of how this molecule is made and how it actually is fluorescent, how that compares with other fluorescent molecules. I have unexpected and I have industry. One of the first talks I gave after the Nobel Prize was to the board of the Woods Hole Marine Biology Lab. And at the end of it, after I gave my talk, one of the members of the board, the president of Zeiss Microscopes in the United States, Jim Sharp, came up to me and he had a little presentation. And he said something startling to me. He said, you know, before 1994, when your paper came out, Zeiss wasn't selling many fluorescent microscopes. and we were going to stop making them. And then your paper came out. So we want to thank you. And I said, and that means you're going to give me a microscope. And he said, no, here's some binoculars. And I'll show you the art thing in a moment. So in my lab, we do ask the question, where are genes expressed? We have a number of genes. They're called MEC, for mechanosensory abnormal. I usually have to explain that although we named the genes MEC, my middle initial is not E. I did not name these after myself. But here's one that's expressed in the touch sensing cells, and here's one that's also needed for touch sensitivity, and it's made in the adjacent muscle. So looking to see where the gene is. We can actually see that this gene, this protein, is actually localized to these dots, which is very important. We're not exactly sure why, but this is the protein that actually does the sensing. We can isolate cells. We can take this animal because we can now see the cells, and we can mutate them and find animals with no cells, more cells, cells in the wrong place, cells that grow out their processes differently, cells that connect in unusual ways. All these things because we can see these. And one of the things that has made seeing these things so good, or one person who's made seeing these things so good, is the third person that shared the Nobel with us, which is Roger Chen. Now, Roger did a lot of things, but he was the first person to come up with a different color fluorescent protein. He also made it much stronger, and then eventually he produced all these wonderful colors, and I'll show one other, I'll probably skip some of the other things that he's done, but he had a number of useful tools that he made from this. So this is only a subset of the many colors. I think it's 15 colors that now spans the rainbow completely, and he got tired of calling them after letters, so they're now all called by fruits. So I think that's blueberry and melon and lime and either lemon or banana, orange and cherry or tomato. I'm not sure which one it is, but a whole bunch of fruits are here, which was easier. And this was used to really great effect by Josh Saines and Jeff Lichtman at Harvard when they wanted to label mouse brain cells. So if you want to look at all of the cells in the central nervous system of a mouse, and you want those cells to all have a different colors throughout the entire spectrum, there's actually only one name you can give this thing, and that's Brainbow. But these are some of their pictures, and the pictures are really strikingly beautiful, really quite wonderful. Let me say a little bit. I'm going to skip some slides because I talk too much, and this is going to go too far. And I will get, come on, there we go. Oh, I have another slide to skip. And I want to get to this slide. A medical application. When we teach undergraduate genetics, we talk about phages that infect bacteria. And one of the things we talk about is a phage infects the bacterium, and the bacterium explodes, and the phage go all over and infect other cells. So the cells lyse. And this is the image we give people of viruses. Now, this would be really good if this happened with every single virus. Because if you could have viruses that were sort of spewed out from one cell onto another, you could make an antibody that would bind to those cells down here, bind to the virus particles. They'd be scarfed up, and that would be the end of them. You'd get rid of it if you were able to, if this is what happened. But when people started to look at HIV and how it was transferred from one cell to another, and I'm going to show you a movie now. Here is a cell that's infected where the HIV proteins are with GFP. Here's a cell that's not infected but about to be by this bolus of virus that goes into the cell. It never went outside. The antibody couldn't touch it. This immediately says, we got to think of a different way to control this. We got to think of a way of these cells interacting with each other so this can't be transferred from one cell to another. By being able to watch these things, all done in mouse cells. Okay, biosensors, other things. You can get GFP linked down to all sorts of things. And Bob Burlage at he was at Oak Ridge National Lab at the time, had an idea. He knew there was a gene that's regulatory part was active in the presence of the explosive TNT. And so he said, what if I take that gene and instead of having what's usually made from it, we'll have it make GFP. So in the presence of TNT, the bacteria will become green. And he made it, and it worked in a dish. It was great. And then because he was at Oak Ridge National Lab and could do this, he had a friend get 5 landmines. Landmines have TNT, and they're known to leak TNT. Now, I should tell you, these are not stupid scientists. These were not connected landmines, but they had the TNT in them. And he had a friend bury them in a 3-meter by 5-meter plot of land. He sprayed the bacteria on top of them, and then came back at night with a handheld UV lamp and got this picture. He was able to find where the landmines were. Now, since landmines are perhaps one of the very worst aspects of war because they kill non-combatants, and after the hostilities have been over, anything that could allow people in a safe way to detect landmines would be really wonderful. Now, apparently the experiment does not work all the time, which is a disaster if you're trying to find landmines, but several groups have been trying to do this. But I bring it up not as a success story so much, but how human ingenuity can apply these things not to do harm, but actually to benefit people in ways that I would never have imagined. But I'm very glad that he has. Other people have done other things. One unexpected thing is a group in Japan has put GFP into silk moths and made GFP-labeled silk moths, and also RFP and so on. And so they've actually made clothing that fluoresces. I almost wore a tie that they made for me, but it's such an ugly, I shouldn't say this on the camera. My wife forbid me from wearing it at the moment. And then I said it was in art. And it has been a little bit in art. I showed you the GFP bunny, Alba. But another thing where it's been used, may be the first example of a human being with GFP. Right? You put this in animals, you got to put the DNA in, right? So it's not really going to be done in people, but there's one person where the claim has been made that they do have the gene for GFP, and that's this guy. If you watch the beginning of the Ang Lee movie Hulk, you will see a jellyfish bounding through the credits. And then this hypodermic syringe comes in and sucks out the GFP. Shimamura should have known about that. And that it's really easy, just take it right out, and then there's a whole little lab notebook. It says bioluminescence and everything. And so the idea is that somehow Bruce Banner, when he was a small baby, got the GFP gene, and it inserted in a gene that gets turned on when he's angry, and so he turns green. Hence the thing. And I actually know the screenwriter for this play, a guy named James Shamus. He's at Columbia in our School of the Arts. And I know him because his daughter and my daughter went to elementary school together. So I went up to him and I said, James, my lab had me see the movie. What a great opening. It's terrific. How did you know about my work? And he looked at me and he says, I have no idea what you do. So I said, no, GFP, remember? And he said, oh, that. We had a guy from MIT working on the set, and he convinced us to do that. I said, I'll take it any way I can. Let me finish up. I'm sorry I'm going to take another 5 minutes or so, but I hope I've convinced you that all those things I said at the beginning are actually not true, that there's lots of exceptions to all of those things. But I also want to say what I do believe I have learned from GFP and the work on GFP. So let me give the list. The first thing I want to say is that scientific success comes by many different routes. I told you about Shimamura's story, which is quite unbelievable. Roger Chen was one of these individuals that had very early promise in science. He was someone, now we call it the Intel Science Prize for high school kids. It used to be called the Westinghouse Science Talent Search Prize. He won it. So he was already doing spectacular, groundbreaking science as a high school student. I was not. Okay. In fact, I'm not going to show you my college grades, especially in chemistry, which if the Nobel Committee had known that, I don't think I would be standing here. It really doesn't make any difference. I think what unites us is not the path we did. There was a time I decided to drop out of science entirely, and I took other jobs for several years. I think it's our passion of what we really like doing and what we like learning about the science that really unites us. And I think that's important. Not what we did as youngsters, it's what we do now. The other thing is that I think many, if not most, discoveries are actually accidental. I told you about Shimamura and throwing the stuff on the floor, but this is not the only case. There's lots of things. In fact, the physicist Enrico Fermi has a wonderful line that goes something on the order of, if you do an experiment and it confirms your hypothesis, you've made a measurement. But if you do an experiment and it doesn't confirm your hypothesis, you've made a discovery. And it's those discoveries that actually push the things forward, certainly much more than the others. Third, I think it's really important to be either ignorant or at least stubborn, and certainly have a willingness to try to do experiments. If we had listened to the people that had worked on GFP and they said there were converting enzymes, we never would have done the experiments. So it was a good idea to ignore them. If it hadn't worked, then we'd maybe think about what the converting enzymes were, but it's really good to ignore it. And this willingness to try. I talked to some students just before this, but I'll say it to everybody. There's something called a weekend experiment. Now, this is an experiment you do Friday night or Saturday morning, Saturday night, Sunday morning, Sunday night, sometime on the weekend. And it's an experiment. The reason you do it on a weekend is no one else is there. And no one else is there because it's a really harebrained idea, but you're really sort of in love with it. And you want to try it, but you're also embarrassed to tell anyone about it. So you come in and you do the experiment on the weekend. If it works, you crow about it on Monday morning. If it doesn't work, you ask people how their weekend went. Now, but it's important to give it a try, to try all these crazy ideas, because every once in a while they work. Not all the time. Most of the time they don't, but they do some. I think that the importance of GFP is not what any of the three of us that got this prize did, or in fact the others like Douglas Prasher, my wife Tula Hazelrig, or Gia Skirkin that contributed to the early work. It could have gone to any of us. Nobel is given to only 3 people. It wasn't even the 6 of us. It's really the thousands of people that have actually added value to GFP because of all the work that they've done. And the importance of GFP is that it has been used by so many people in so many wonderful and novel ways. And so I'd like to think of GFP as a metaphor for how we do science. Because just as GFP absorbs light of one wavelength and converts it to light of another wavelength that is given off, scientists, we take in the ideas and observations of other people, we modify them by our own thoughts and experiments, and then we give them off to others to change in turn. So it's a constant bringing things in and giving them out and seeing them changed by other people as well. And this cumulative effect of what's happened that made GFP so important. People have asked me, why was GFP given the prize in chemistry? Why wasn't it given a prize in medicine physiology? And I think it's because they gave the prize to the molecule. The 3 of us came along for the ride, which was a nice ride. But I think it was because of the molecule and the importance the molecule had. This looks like it's an advertisement. University and grant support was essential. Let me say why I have this on the slide. University support. Columbia University could care less what I do. They give me the freedom to do any experiment I want. And that freedom I enjoy a lot. it let me do this experiment with GFP that, didn't ask anyone's permission. We just did it. We didn't have any grant support for it either. We just used our existing grant money to do it. But having that freedom, if it succeeds, great. If it doesn't, that's too bad. But having that freedom to do it has been exceptionally important to me. Grant support, and I bring this up because I think non-scientists often don't realize what grant support is. What it is not is a contract. I don't write a proposal that says, here are the 4 things that I want to do, and if you think those are good experiments, pay me, and I promise to do those 4 experiments. That's a contract. You're saying what you're going to do, and you get paid when you do that. But we don't write contracts. What we write instead is a grant proposal. The grant proposal says, at this point in time, the 4 most important experiments that I can do are these. And if you think those are right, support me. And then there's this unwritten part in all the grant proposals. It's always assumed. And that unwritten part reads, if a better idea comes to me, I'm going to use the money for that. Or if there's a better way of looking at the cells, if GFP is invented, or CRISPR-Cas9, or other things are done so that I can use those ideas and get to the results, I would be an idiot to stick with the old method. I'm going to use the new method. I'm going to use the new ideas. I'm going to go after what's more exciting. And when it comes up for renewal, you're going to tell me whether I was a complete idiot or not on the basis of what I've accomplished. I don't think anyone knows what they're going to be doing more than maybe 3 months in the future. And we write grants for 4 and 5 years. Everybody knows we're lying. That's on camera now too. Okay, good. The people that really drive the lab are the students and the postdocs. When we started giving GFP away, it was the heads of the lab that called me up. And when they called me up, they would always start their sentences, their call, in the same way. They'd start off and say, my student or my postdoc told me that you have something called GFP. Tell me what it is and why do they want it? And I'd tell them and they'd say, absolutely. We want this. This is great. Yes, but it was the graduate students, the undergraduates, and the postdocs that actually pushed the science because they were excited about what was happening. That still happens in my lab all the time. Next to last point. This all came about because Shimomura was interested in a fundamental problem of biology. How is it that some organisms can generate light? And along the way, he makes this accidental discovery about a fluorescent protein, something that no one had imagined. It points to the fact that we should not narrow our views to only do studies on what we now call model organisms, which are all useful. C. elegans was not a model organism when I started, and now it's become a model organism. But by working on non-traditional organisms, we're going to learn a lot more about biology, and that's going to help everybody. And the final point actually draws from that, which is that fundamental research, basic research, is really essential. There's a big push in the United States now for what's called translational research, to translate what is found in the laboratory to cures and preventive measures in the clinics. It sells really well. Unfortunately, just about everybody you'll ever talk to in science will admit that if you want good translational research, and as I got older, I want good translational research, I think that's important, but in order for that to happen, you have to have something to translate. Many people have made this point, that we have to have the ability to feed new information into the system so that it can be translated. And we are learning about things on a virtually daily basis that we had no inkling about before. And to say, okay, we've learned enough, now we're just going to translate, is really missing the whole point, is that we really need the fundamental research. We can't predict what the fundamental research is. When Charles Towne developed the laser, in the 1950s, he didn't have record companies and movie companies coming to him saying, CDs and DVDs, this is great. We can use a laser for that. Didn't have medical people coming and saying, we can do operations with a laser. None of that. It took years for these things to develop from the basic physics that he had come up with, and there's lots of it. And I want to leave you with my favorite quote about basic or fundamental research. It was said by Robert Wilson, who was the first head of the Fermi Lab in Batavia, Illinois. The Fermi Lab, until the CERN super collider came online a couple years ago, it was the biggest particle accelerator in the world. It was built after a meeting of a joint congressional committee, and actually a bill was passed in 1969. And Wilson was asked to come to that committee by Rhode Island Senator John Pastore, and he was asked, we'd like you to come and testify and show people why it's so important for the US government to pay for this science project. And so he went, and Pastore tried to help him by asking him what he thought was an easy question that would be really good for the other Congress people to hear about. And that question was, Dr. Wilson, can you tell us how many ways, remember it's 1969, middle of the Vietnam War, a lot of concern about safety and defense and so on. He says, how many ways will this accelerator help national security? And Wilson said, none. Not the answer Pastore was looking for. He asked it again, none. Finally he said, no, look, in what respect will this accelerator help the national defense? At which point, Robert R. Wilson said, it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much.